Hi everyone, Anna is here, and in this video we will be discussing the biology behind the dissociation curves. By the end of this video you will be able to discuss the components of dissociation curves and how they shift to the left and to the right and what are the factors affecting this. So please grab a piece of paper so you can draw alongside with me and take your notes. And without further ado, let's just jump into this video. The only thing I ask in return is please press like the video and leave the comment below of what other video you'd like to see in my A-Level Biology Masterclass, and let's just jump into it. To understand this topic of dissociation curves, I suggest we start by separating and dividing it into separate components, starting with hemoglobin. So a reminder, hemoglobin is a quaternary protein consisting of four subunits, four polypeptide subunits, and in turn one of these subunits contains a heme group that is able to bind to an oxygen molecule. And in this equation I'm drawing at the moment, so hemoglobin in total therefore can bind to four separate oxygen molecules, making hemoglobin O8, because remember oxygen consists of two oxygen atoms. So we have the hemoglobin here, and then when it's bound fully to oxygen we refer to it as oxyhemoglobin, and this reaction is reversible. Furthermore, when hemoglobin binds to oxygen, we refer to this process as loading, and then when hemoglobin dissociates from oxygen, we refer to it as unloading, and we will use this terminology for the rest of the video. So the second component of dissociation curves, or a term that you need to know, is affinity for oxygen. Affinity for oxygen is tendency of a molecule to bind to oxygen, and again we will be using this terminology throughout the video. The third component referred to this dissociation curve is partial pressure, and specifically it's going to be referred to partial pressure of oxygen. So when we refer to oxygen, we can't really say concentration of oxygen in the atmosphere, but instead we refer to it as partial pressure, which means a measure of oxygen concentration. If you guys learn these definitions and concepts, then we can go ahead and really get into the dissociation curves and draw some examples out. So at this point, I want you to draw a graph. So I would recommend take a half a page and draw it big, and so we could then label it as we go together. So if you guys draw the graph, on the x-axis we're going to have the partial oxygen pressure, which is represented by little p as shown in this video. And then on the y-axis we're going to have percentage saturation of hemoglobin with oxygen. So basically, in simple terms, how many oxygen molecules are binding to hemoglobin. And I want you to draw your dissociation curve as an S or sigmoid shape. One of the skills that you're required to know is to be able to explain each part of the curve and why it's its specific shape. So how does the gradient change as we go across of this curve? Okay, so I'm going to start labeling parts of the curve with hemoglobin and how many oxygen molecules it's bound to. So we start with hemoglobin O2, i.e. one molecule, hemoglobin O4, 2 oxygen molecules, hemoglobin O6, 3 molecules, and hemoglobin O8, 4 molecules. As remember that oxygen exists as O2 molecule in the environment. Okay, so the reason it is this shape. So if we start with hemoglobin O2 at the bottom, we can see that the gradient is actually slightly less steep. The reason is that hemoglobin, as a reminder, it consists of 4 subunits. So when one oxygen binds to that subunit, it actually is less favorable at the beginning because the hemoglobin is going to be in a slightly unfavorable conformational change, or the shape as you can refer to it, and so therefore it's hard for the first molecule of oxygen to bind to it. If we then copy and just basically draw the same diagram for hemoglobin or O4, and we just add the second oxygen, then we will add an extra oxygen to hemoglobin O6, and then to hemoglobin O8 we're going to add the fourth oxygen. But as you can see, the gradient keeps changing across the dissociation curve, and it gets less steep, particularly for hemoglobin O8. The reason for that is that the oxygen at this point is less likely to find that fourth empty subunit to bind, just by chance. As you can see, I can show here with an arrow, so that the oxygen has to actually bind at the correct angle, so it's less likely, so therefore the gradient of the dissociation curve at that point is represented lower, but it's quite steep for hemoglobin O6 and hemoglobin O4. So now guys, Let's look at different factors that affect dissociation curves. Topic quite often comes in exams. So there are certain conditions that will change whether the dissociation curve will be shifted to the left and to the right, and that will represent the affinity of hemoglobin for oxygen. So how strongly can it bind to oxygen? Okay, so if we go through factor number 1, we're going to call it low oxygen environment. So how does the dissociation curve change if suddenly the partial pressure of oxygen goes lower? So an example that you might think of is basically if we take human as an example of a mammalian organism, and let's compare it to a lugworm. Lugworms is a classic A-level biology example of an organism that lives on the beaches in the sand, and it's basically lives in the low oxygen environment because the water obviously with the tide goes up and down, so the lugworm doesn't get much oxygen. So the human sigmoid curve, the dissociation curve, are drawn it like that. And then if in this case you can imagine the lugworm, the hemoglobin, it needs to be able to have a higher affinity for oxygen, because if it's not exposed to much oxygen, it needs to be able to almost like grab onto it tighter. So therefore, this is represented by a dissociation curve with a shift to the left. So in this case, lugworm has hemoglobin that in turn has a higher affinity for oxygen, and then this can be compared to human, and then I would just suggest, guys, you note this down, that in human, hemoglobin has lower affinity for oxygen compared to the lugworm from this graph. These examples of graphs come up in exams quite a lot, so I would really recommend learning this phrasing, and they usually have like 2 or 3 marker questions associated with these particular graphs, and students do tend to lose quite a lot of marks, so the wording here is very important. Let's now look at the second case of where there is low oxygen environment. So for some exam boards, you need guys to know the difference between the adult and fetal hemoglobin. Fetal hemoglobin will have a higher affinity for oxygen, as fetus is exposed to less oxygen compared to adults. And so therefore, as depicted here by the arrow, the hemoglobin dissociation curve in this case will be shifted to the left. Okay, the second factor that can affect the dissociation curves is the high activity levels. So again, guys, if you could just draw another graph for me, please, and we're going to be comparing the dissociation curve for the human hemoglobin versus hawk's hemoglobin. A hawk is a bird that obviously flies in the sky, and it actually uses a lot of energy. So in this case, hawk's hemoglobin needs to be able to release oxygen more readily compared to human hemoglobin, because remember, oxygen is going to be used for the aerobic respiration, so then the energy can be released in a form of ATP for various cellular processes. So in this case, the hawk's dissociation curve for the hemoglobin is going to be shifted to the right, as depicted by the arrow here again. Third factor that can affect the dissociation curve is the size, in particular the surface area and to volume ratio. So what I want you to do is draw a graph and draw 2 dissociation curves for human versus mouse. Pause the video and ask yourself, how does the hemoglobin's affinity for oxygen change with the size versus human and mouse? Again, quite a common exam question, and this is the way to answer it. So the mouse has a higher surface area to volume ratio, so therefore it loses heat quicker. It has a higher metabolic rate, and therefore a higher oxygen demand, and therefore the dissociation curve shifts to the right, suggesting that hemoglobin has a lower affinity for oxygen compared to human. Oxygen then can be unloaded much faster and more readily. So these are the examples of dissociation curves that can come up in exams. I hope this video really simplified this concept. In the next video, I'm going to release and discuss the Bohr effect and chloride shift. Please like this video, subscribe to my channel, and please do let me know in the comments below what video you want me to make next. And then I'll see you in my next video. Goodbye.